Hello everyone, welcome back to Biophilia, your all-time study mate. Today we are going to start a new chapter about biotechnology principle and processes. If you like this video, then don't forget to like, share and subscribe to our amazing channel Biophilia. So here we are to understand a different field of biology that is about biotechnology. Biotechnology is referring to use of living organisms or their products to modify human health and human environment. So it might be regarding the in vitro fertilization, it may be the recent vaccine production, or even the gene correction. Now, how biotechnology is used in these various fields will be studied in the future few minutes. So there is a body that is European Federation of Biotechnology that is defining biotechnology as integration of natural science and organism, cells, parts, therefore and molecular analogs for products and services. So biotechnology is going to be the upcoming field wherein many modifications, a lot of human intervention is being carried out in the animal and the plants as well. So let us see in detail about how and what are the principles of biotechnology. So principles, there are two major core techniques on which the entire biotechnology is based on. So for the birth of the modern biotechnology that majorly is being followed nowadays, it's the first, that is genetic engineering. So genetic engineering, the word can specify the meaning of it, as genetic is regarding the genes that your body has. So generally when we speak about human intervention, it is regarding the plant or the animal body. So majorly you would be dealing with the DNA. Rarely, in some cases, you would be targeting RNA, but majorly it's the DNA that we are speaking about. Engineering, that means we are making some modifications in the genetic material, that is DNA. So if there is any sort of alteration in the chemistry of genetic material, and what alterations, you are introducing some new phenotypes. You are changing the phenotype, you are changing how the organism looks, how it behaves, or how does it produce. If you are changing that sort of thing with the help of the genes. Okay, if once you change the genes, it is going to be carried out from the parent to the progeny, because parent से progeny क्या travel करेगा, from parent to progeny only the genes are going to be traveling. The traveling part is the gene. If you modify the gene, then you can modify the progeny as well. क्योंकि अगर सिर्फ parent को ही change किया तो वो generation तक ही सीमित रहेगा. But if you want the changes in the child as well, then the gene should be targeted, and for that reason the modified gene or the modified DNA, as we are majorly speaking about DNA, the recombinant DNA, the recombined, because you are changing the chemistry of the genetic material, so that will be called as rDNA. And this rDNA is going to have different properties, जिसे आप जिस हिसाब से उसे modify करोगे, the same type of changes will be occur in the recombinant DNA. That is the first thing, that you are altering the chemistry. Only one the genetic material is changed and you obtain your desired recombinant DNA. In the second part, we see the bioprocess engineering, wherein you have to maintain the sterile ambience, क्योंकि जो आपने genetic engineering से एक DNA को change किया है, that DNA needs to be multiplied and it should be commercialized. So these engineering processes are used wherein you are going to select any sort of desired microbe or eukaryotic cell, और ये eukaryotic cells के अंदर you are going to manufacture these things like antibodies or vaccines or enzymes which you are creating from the genetic engineering. So एक के अंदर चीजें आप genetic engineering से करोगे और उसके multiple copies उनका production के लिए you are going to use bioprocess engineering. So that is commercialization, but the basic fundamental changes will be done through the principle of genetic engineering. Next we see is how are the techniques of this genetic engineering. एक single DNA को change करने के लिए what all things do you need to proceed with? So the first thing you do is to introduce the new gene, या वो gene जो आपको नहीं चाहिए, you remove it. Either it is based on addition or it is based on subtraction of the gene that you don't want. So if you want to add something, then you would take the external DNA, take a source that is desirable to you, and inject it into some other DNA. If you don't want it, then also we will see different kinds of methods wherein you can understand how the genes are added and how they are subtracted. So first we need to see about gene cloning, what is it and how does it occur. Second is creation of recombinant DNA. जो changes हम कर रहे हैं DNA में, it is not like that you cut it from here and then you are going to just paste some other problem. It is the DNA small part that you cannot see with your naked eye as well. In that you have to make changes. So how is it done? We have to see that as well. And then the gene transfer, whatever has been created by you, it needs to be posted into a host, and this host should accept it. It is not always that the cell that you are taking the genetic recombinant DNA into, it will be accepted by the host, because it is new to the host. It will not accept. Then what are the various ways to which the host will accept these changes, and that will help you to manufacture. So all these things need to be taken care of when you are undergoing the genetic engineering. So let us start with first the steps of gene cloning. So gene cloning, 5 basic steps to be understood. But before that, what is gene? You know, what is cloning? Cloning is nothing but making of multiple identical copies. Now any template DNA, suppose you have changed a particular DNA, और उसको चेंज करने के बाद यू वांट टू मेक द कॉपीज, बिकॉज एक ही को चेंज करके यू आर नॉट गोइंग टू हैव एनी देयर इज नो एडवांटेज ऑफ इट. बट इफ यू हैव मल्टीपल कॉपीज, then it is helpful for you to multiply it. But if what if you repeat the same process that you have done for the first one, it would be taking a lot of time. But instead of time, you need to work smart. So for that smartness, you need gene cloning. यानी आपने एक में changes किए, that took you months together, and after this you are going to introduce this particular changed DNA into some kind of a bacteria, or you might be introducing it to some kind of a eukaryotic cell. और ये cells या ये host जो होंगे, what they will do, they will have rapid division, and because of the rapid division, in few hours you are going to get multiple number of this recombined DNA. So for that, first of all, you need to clone the gene. और कौन सा gene clone करना है? The recombinant one, the changed one, the modified one. So first of all, you need to construct that. आपको एक जो original gene है, या you say the wild type is there, wild type के अंदर you need to insert, or you need to change the desired gene. And after the wild type and the desired type has been mixed, you will get a recombinant DNA molecule. Now this recombinant DNA molecule should be transported into the host. Now for example, the host is a bacteria. Now why would you choose a bacteria? Because bacteria would have, it is smaller in size, so you can culture bacteria, large amount of bacteria in small amount of place, and even the rapid division is possible, so that in minimum time you will have major quantity of the recombined DNA molecule. So host cell, bacteria is chosen. Third step, you need to multiply. So when will the multiplication of the recombinant DNA occur? Only when the bacteria will divide. So bacteria, जब एक बैक्टीरिया से सेल डिवीजन होके दो बैक्टीरिया बनेंगे, दो से जब चार होंगे, so that is nothing but along with it the recombinant DNA is also getting multiplied. So we want this, that the host cell should multiply. So all the favorable conditions should be provided so that the host cell can multiply. After the host cell has been multiplied, you will get the number of divisions, and the divided colonies would be nothing but the clone. So that is the basic 5 steps about gene cloning. The next we see that how was the first step, that is making of the recombinant DNA. DNA के अंदर changes जो किए, how it was possible. So for the first time when the recombined DNA was formed, it was possible because of the antibiotic resistance which was present in the plasmid. Now what is plasmid? Plasmid is the extracellular, extranuclear material that is present majorly in the bacteria. So which bacteria was it used? It was Salmonella typhimurium. So this bacteria was having the wild type antibody resistance. So using this, there were two scientists, Stanley Colin and Herbert Boyer. In 1972, they were able to cut this antibiotic resistance gene. कहाँ से cut किया? They had taken their desired gene from Salmonella, and once they have cut the desired gene from Salmonella, they have attached it to the other bacteria, other host. So Salmonella कौन हो गया? Salmonella was the donor. It was the bacteria which has the desired gene, but it was transferred by a plasmid into Escherichia coli. So Escherichia coli has become the host bacteria. So Escherichia coli के अंदर जब भी divisions होंगे, it would replicate and the DNA polymerase enzymes would make multiple copies of the desired gene, that was the antibiotic resistance gene. So how are the basic steps? अब ये उन्होंने किया, but if you want to do it in future, then there are basic 3 steps that should be taken care of when you are forming any sort of genetically modified organism. The first one would be that to identify the DNA of the desirable genes. For example, if you want the antibiotic resistance, so in the long chain of DNA, you need to see that which is the part that you desire. You don't want the entire DNA to be transported into a new cell, so you just want a small part of it. The part which is desired to you, that should be recognized. और जैसे ही ये recognize हो जाएगा, now what you want to do, you have to detach it from the whole and you need to transport it. So you need to introduce it to the host cell. Now host cell को introduce करने के लिए first you need to separate it. So how are you going to separate the desired gene? क्योंकि बाकी का तो waste है आपके लिए, you don't need the other genes, you just need the part that you wish to introduce. So for that you need to cut, you need to separate. कट करने के बाद नए host में introduce करने के लिए you also need to paste it somewhere. Now to paste it, you need to stick it together to other DNA. Now how is this all going to happen? So ये जो cutting है, pasting है, to introduce it to the new host, and then the last step comes where you need to maintain. जो भी सब किया है वो एक ही cell पे नहीं करना है. You have to continue to the different progenies. So you also have to maintain that particular changes that you have made. So with the identification, introduction and maintenance, you can modify any sort of organism. It looks simple just to maintain it in 3 steps, but how is all that thing be done in real life? So desired gene को identify करने के बाद, once you need to separate it from that DNA, you need to cut it, and that cutting of the DNA is going to be done by molecular scissors, which is called as restriction enzymes. Restriction enzymes are going to be the molecular scissors, and these scissors are going to be kind of आपको जो cut करना है only that will be cut, others will be kept as it is. अब ये जो कट किया है उसको आपको ट्रांसपोर्ट करना है to the desired location. Now how would you cannot just pick it up with a forceps and just transport it to the other cell. You can't do that because it is so tiny it would break. Now what to do? You cannot transport it by your own hands. You need a vehicle. Now that vehicle would be nothing but a plasmid DNA. This plasmid DNA is going to act as a vector, as a vehicle. This is a vehicle. समझो कोई कार है, बस है, जिसके अंदर ये passenger यानी your desired gene will be sitting inside, and once it sits inside, what will be done? It will be transported to the desired host cell. Now plasmid ने transport कर दिया, but it needs to be attached to the host cell genome so that it can get multiplied along with the host cell. So for that purpose, to get mixed up with the host cell DNA, you need to paste it to the new DNA. So for the pasting purpose, you need DNA ligase. This DNA ligase is going to paste it, and along with it, that is the molecular glue. Molecular glue is DNA ligase. Molecular scissors are going to be restriction enzymes. Now all these are just the enzymes which are going to be named as scissors and glue. And afterwards, once it is pasted with the DNA ligase as the molecular glue, whatever the new structure has been formed will be called as a recombinant DNA. So recombinant DNA कैसे दिखेगा? this was the host DNA. Okay, this was the original, the wild type. Now what you have added? You have added the desired one, and then the part will be modified somewhere wherever it gets pasted. So that will be nothing but your recombinant DNA. So this recombinant DNA now needs to be done in a specific ways. Now these were the tools. ये तो कुछ tools use किए हमने, that were the scissors and the glue. But how in detail is it performed? Let us see with the different tools of recombination. So tools of recombination, major three चीजें चाहिए हमें. First of all, you need the enzymes. Therein you can modify the DNA of the desired person. So desired के लिए सबसे पहले restriction enzymes चाहिए. That will be your scissors to cut it from the cell जहाँ पे आपको ये चीज मिली है. वहां से कट करने के बाद you need to multiply that number. So for multiplication to make into multiple copies, you need to have polymerase, and ligase will be the glue which will paste it to the host cell. Now vectors will be the vehicles which will transport it to the host cell, and host cell जो होगा that is going to multiply to give progenies. So progeny production के लिए you need the host cell. Now with the first, that is restriction enzymes, these are the chemical knives which are going to cut the DNA fragment. अब जो first restriction enzyme को identify किया था, that was the restriction endonuclease. Endonuclease means it cuts the DNA in center. Endo means inner cut will be given, whereas exonucleases are also present. So endonucleases, the first name, this name is important, that is hind 2, which was used to cut DNA molecules at a specific point. अब ये specific point कैसे choose होगा? There are so many base pairs, that is A हो गया, T, C, G, T, A, C, G. There are many such number of base pairing, but where to cut, that is a point. So these restriction endonucleases have specific sites which are allotted to them. So for the case of hind 2, it will recognize the 6 base pairs, and these 6 pairs, once they are identified, यानी scanning होगी लंबे से DNA में, there will be scanning of this endonuclease, and after the scanning is done, और एक part आएगा wherein it will find its particular desired zone of 6 base pairs. जैसे ही उसको 6 base pairs मिल जाएंगे, it will cut into that particular location. So these restriction enzymes are majorly being obtained only from prokaryotes. These are not found in eukaryotes. Restriction enzymes prokaryotes में मिलते हैं. Now this is a reason by why it is not found in the eukaryotes, because restriction enzymes are as natural defense for the bacteriophage infection. जैसे अगर यूकैरियोटिक सेल है, दिस इज अ यूकैरियोटिक सेल. यूकैरियोटिक सेल के अंदर इफ अ वायरस इज एंटरिंग, you would call it as the virus infection or viral infection. But what if it is entering into a bacteria? अगर वो बैक्टीरिया के अंदर वायरस घुस रहा है, then that virus would be called as a bacteriophage, because it is entering into a microscopic field only. So virus entering into the bacteria is called as bacteriophage. So this bacteriophages needs to be, जैसे human cells, eukaryotic cells will prevent the viral infection. Similarly, bacterial cells will have their own defense in the form of restriction enzymes to prevent the bacteriophage infection. So how are these restriction enzymes been given a name? Hind 2 हमने नाम देखा, how was it given? So for example, if you consider about EcoR1, what is it? EcoR1. So eco is the term. कैसे याद रखें? Eco क्या है? R क्या है? 1 क्या है? So eco is coming from Escherichia coli. That means from where it is derived. From where have you obtained this restriction enzyme? So we have seen it is obtaining from the prokaryotes. So eco is referring to the Escherichia coli. फिर R लिखा है. R कहाँ है? R is nothing but the strain, that is, it is either the smooth strain or the rough strain. So it was taken from the rough strain. So rough का R लिख दिया. And then it was indicating कि ये सबसे पहले निकाला था, that was the first one to be identified from that particular strain of bacteria. So you have given the Roman number. 1. So eco R1 may say eco is for E. coli, R is for the rough strain, and 1 is the order in which this enzyme was obtained. So in this way, you are going to name the different kinds of restriction enzymes. So हो सकता है in some other one you might find the strain has been changed or strain है नहीं तो then like hind 2 you won't find any sort of other letter. Now, how is the working of this restriction enzyme? As I told you that there would be scanning, and then when the desired type of base pairs are located, then they would cut that particular thing. Now, how does this thing occur? So first of all, it will recognize a palindromic nucleotide sequence. Now, what do you understand by a palindromic nucleotide sequence? Palindromic means once you read it from left to right or you read it from right to left, it will be the same. How is it possible? Just remember about the word that you must have seen, that is madam. M-A-D-A-M. You read it from left to right or you read it right to left. You would spell the same thing. So this is nothing but called as a palindromic sequence. Same kind of thing once it is found in the DNA, वैसी जगहों पे जाके ये restriction enzyme जाके कट करेगा. अब कट करेगा, what is it going to cut exactly? It will be breaking the sugar phosphate backbones. कट करेगा मतलब DNA को कट करना is nothing but breaking the bonds between them. So double helix के बीच में जो sugar phosphate bonds है, those will be cut. Now this cutting fashion is also seen in two types. Either one कि वो एक zigzag manner में cut करेगा या it would cut straight. Now if it is cutting in a zigzag manner, so zigzag manner में cut करेगा, so what it will give you? It will give you a sticky, a cohesive or a staggered end. Okay, it would give you a sticky, cohesive or a staggered end. Now what is the advantage of sticky, staggered or cohesive end is it would be as a zigzag puzzle. Zigzag puzzle होगा, it would fit in the particular host cell genome very perfectly. अब perfectly fit होने के लिए it needs to be a sticky end. So sticky ends या staggered ends cut करने वाले restriction enzyme का एक example will be EcoR1. EcoR1 is going to be providing you the sticky or the cohesive ends. Now blunt end would be once the bonds are broken straight up and down. So straight bonds cut होंगे, that will give you a blunt or a flush end. Yani you can see the difference about how the ends are created. These ends in the sticky ends are in zigzag manner, but in blunt it is straight lines. So straight lines को combine करने में या to make a bond between them is much more difficult, and it won't be it won't be that much effective as compared to the sticky ones. But sometimes you need blunt ends, so you can use the restriction enzyme as SAMA1, or for majorly use, that is the sticky ends, EcoR1 is one of the examples that is providing sticky ends. Next we see is the DNA ligases. Again, it is a part of enzyme that is manipulating the DNA. अब ये कैसे मैनिपुलेट करेगा? इट इज द मॉलिक्यूलर ग्लू. दिस मॉलिक्यूलर ग्लू, जैसे हमने जब मॉलिक्यूलर सीजर्स बोला, सो सीजर्स वर डूइंग व्हाट? द सीजर्स वर ब्रेकिंग द बॉन्ड. नाउ व्हाट इज द ग्लू गोइंग टू डू? Glue is going to form the bond, or it will make the bond. So the bonds that are formed are going to be phosphodiester bonds between the adjacent nucleotides, and the covalent links will be provided between the two fragments of the double stranded DNA. अब ये सब जो करना है, नए bonds form करने हैं, so any formation needs energy. So energy will be utilized from the host cell to form these bonds. Now majorly what are the molecular glues or what are the enzymes which are oftenly used for this DNA ligation? So it is the T4 DNA ligase. अब T4 कहाँ से आया है? T4 is regarding that it is obtained from the bacteriophage, that is T4. So the T4 DNA ligase is one of the example for the DNA ligases, that is nothing but molecular glue. अब हमने cut किया, paste किया, now we want to increase its quantity. So DNA polymerases. These DNA polymerases are going to synthesize a new strand which will be complementary to that. So this complementary strand will be formed from 3 prime to 5 prime, और already 5 prime to 3 prime का template बना हुआ है. Using that template, the complementary strand will be provided, and one of the example which is generally being used, that is DNA polymerase 1. So for the molecular scissors, you have examples like Hind2, which was the first restriction enzyme. One more example is EcoR1. Second example is SAMA1. For molecular glues, that is DNAligases, you have T4 DNA ligase, and polymerases, DNA polymerase 1. So using these enzymes that manipulate DNA, you can change the DNA sequences, and after changing, you need to transport it to the host cell. So for the transportation, you need some vehicle, the vector. So this vector is nothing but a carrier which is going to deliver the foreign piece of DNA to the host organism. And once the host organism के अंदर वो जाएगा, it will release this, and that means taxi system है. What is it? It is just a taxi system which will pick up and provide the drop service. So pick up and drop हो गया. Pick up कहाँ से किया? Pickup was from the place wherein you have made this genetic changes, and drop will be into the host cell. So after the pickup and drop, how is it going to sit into this taxi? Where is the taxi space? So that seat is going to be somewhere, depending upon what sort of vector you are choosing. Now once you are choosing a vector, why do you need a vector? First of all, it is easy to link the foreign DNA, यानी ये taxi system जो है, it is easy to get, availability is good, so you can have the better transport system. Selection of recombinants and non-recombinants can be done, यानी सही में ये taxi ने, ये vector ने host cell के अंदर पहुंचाया है कि नहीं, that can be checked. So based on that, you need to choose a cloning vector. So there are different kinds of cloning vectors that we will see, and what are the different sites wherein the cloning vectors have, that also we will see in detail. So special features यानी कुछ sites होते हैं, there are some positions in a vector that should be there which are useful, and what are their uses? Let us see one by one. So first is the use, that is origin of replication. As the name indicates, it will be the start place wherein the replication is going to occur. So any piece of DNA which needs to be multiplied in the number that is referred in the host organism, तो उसको कहाँ पे रखना चाहिए? You should keep that foreign piece of DNA wherever there is the origin of replication. For example, in this particular diagram, we have the origin. Where do we have the origin of replication? We have it over here. If you can see, this is the origin of replication. Origin of replication के site पे अगर we have placed the recombinant DNA, then what will happen? Since it is the start of replication, any DNA that we are going to place is going to get replicated. कहीं और रखा तो हो सकता है that particular site is not multiplied with the progeny, but since it is the origin, it needs to start from there, and that is the reason the alien DNA, that is the DNA that you are going to modify, needs to be placed onto this origin of replication. The second important feature that a vector should have is selection marker. यानी आपके पास कुछ ऐसी टेक्नोलॉजी होनी चाहिए जिससे पता चले कि आपके टैक्सी ने पैसेंजर को सही जगह पे पहुंचाया है कि नहीं. So that system is nothing but the selection marker. So how will you identify if your particular gene of interest has been provided, or it has been settled into the vector or not? So for that, you have selection markers. Say for example, you have resistant genes in the case of antibiotics like ampicillin or tetracycline. These antibiotic, if you grow that particular vector in the antibiotic, if it grows, then say for example that antibiotic resistance is not there, and if it fails to grow, यानी the changes has been made and जो resistance था वो निकल गया है, and once you can confirm कि resistance अब नहीं बचा है, then you can say that now the gene of interest has been settled into that particular area. Now ampicillin में या tetracycline में करना is difficult because you need two plates at simultaneous positions. But instead of that, there are some other ways as well. That is the gene that is LAC-Z, which is encoding for beta-galactosidase. Now this beta-galactosidase is a chromogenic gene. Chromogenic means it will produce color. So blue color production अगर होता है, that means that the gene is not modified. क्योंकि normal जो gene है, normal gene जो भी होगा, what it will do, it will produce blue color. Now if you modify करने के बाद what should happen? Modify करने के बाद blue color production should stop because you have removed that gene. और अगर जो gene निकल गया है, it should not produce the color. So if you have removed it, if you have modified it, then it should have colorless colonies. So bacterial colonies जब भी grow होंगे, you just have to select the ones which are colorless. Color ones are the wanted or the desired colonies. Blue वाले को आप क्या करोगे? You won't use it, you will throw it away. Then the cloning sites. Cloning site means there are some places wherein you need to have multiple genes to be set up in a single bacteria. So वहाँ पे cloning sites use होंगे wherein many restriction enzymes will be used. And for that important reason, one point to be remembered is cloning sites जो होंगे, that cloning sites should not have the sites which are common to many restriction enzymes. क्योंकि क्या होगा, फॉर एग्जाम्पल इफ यू हैव अ वेक्टर जिसके क्लोनिंग साइट्स से फॉर एग्जाम्पल सिंगल काइंड ऑफ अ क्लोनिंग साइट है विच आर इन मल्टीपल लोकेशन, ओके, देर आर मेनी सेम लोकेशंस, और दूसरा एक है द क्लोनिंग वेक्टर जिसमें यू हैव ओनली फ्यू क्लोनिंग साइट्स. तो क्लोनिंग साइट कम होने की वजह से क्या होगा, द रिस्ट्रिक्शन एंजाइम विल कट इनटू few segments. But if there are multiple cloning sites, then what will happen? Cutting होगी, that cutting will be at multiple places, and multiple places पे जब भी cutting होगी, it would destruct the DNA, and that is not desirable. So for that reason, you should not have multiple few recognized sites जो fragments को complicate ना करे during the gene cloning. So there are some other site as well जो बहुत सारे पास में चीजें होती है, that would be called as the multiple cloning sites, but generally to be taken care कि अगर आप एक restriction enzyme use कर रहे हो, that should be having the recognizing site at a very far distances so that you do not have many fragments cut. Final thing to remember about the vector is उसका size. आपने सारे प्रॉपर्टीज देख लिए, ओरिजिन देख लिया, उसका सिलेक्शन देख लिया, यू हैव सीन वेयर नीड्स टू बी कट, एवरीथिंग हैज बीन सेटल्ड. बट अगर साइज ही बहुत बड़ा है, देन वॉट एवर चेंजेस यू मेक, those changes won't be passed on to a road which is not able to handle that big car. For example, जैसे हमारी टैक्सी है, taxi is your vector. Say for example there is a big road, okay, this is a big road, ये big road के ऊपर आपकी छोटी सी टैक्सी है, आराम से निकल जाएगी. But what if your road is the same way but your taxi is very big? एक बड़ी सी टैक्सी लेकर आप जा रहे हो, एक बड़ी बस लेकर जा रहे हो छोटी सी गलियों में, सिटी की गलियों में, is it possible that your big vehicle will enter into that small roads? It won't enter. The same thing is, once you have a small vector, that small vector will have a greater tendency to enter into the host cell, and it will also not have the tendency to break down. क्योंकि बड़ी चीजें है, when you are processing it further, हो सकता है बड़ी चीजें processing में कट फूट जाए, डैमेज हो जाए. once you are trying to enter it into any छोटी गलियों से जा रहे हो, पता चला आपके शीशे को स्क्रैच हो गया, शीशा नीचे गिर गया, anything can happen. So that thing should not happen. So the size of the vector that you would choose, it should be small enough to enter and not to break down during the further purification procedure. So these are some initial factors that you need to understand while selecting a vector. The next is the example of such kind of a vector. Vectors can be different types, but one of the important one that you should know is the plasmid. A plasmids क्या होते हैं? We have come across this term previously as well. These are the extra chromosomal material. Because these are extra chromosomal found in the majorly the bacteria, so वो बैक्टीरियल सेल के अंदर का जो भी जेनेटिक्स है वो चेंज नहीं करेगा. इट इज द एक्स्ट्रा क्रोमोसोमल. उसकी लाइफ के रिलेटेड जो भी जीन्स है, यू आर नॉट चेंजिंग दोस. दिस इज एक्स्ट्रा फॉर दैट पर्टिकुलर ऑर्गेनिज्म where you are making the changes. So how can you identify this extra chromosomal material? It will be outside the nucleus. It will be circular and it is not essential, very essential for the bacteria. It will be double-stranded, autonomous and self-replicating pieces of DNA. And because of it is self-replicating, you can use it to have multiple copies as well. So what is the major property जिसकी वजह से you are choosing the plasmids as well? Because it has the property of antibiotic resistance as well as virulence. So that two characteristics, these two properties can be used by the geneticist so that the plasmids are helpful for the genetic revolution. So there is one example of the plasmid that we generally use, that is PBR322. PBR322 will have different sites in its vector. That means there will be a ORI, that is origin of replication. After origin of replication, there are different sites wherein the restriction endonucleases can cut. For example, EcoR1, that is one of the endonuclease we have seen. There is HIND2, likewise we have a HIND3. This is also the site which is recognized that particular endonuclease. So EcoR1 की जो site है, that will be the site that this particular endonuclease will come and cut at that particular position. So that is the EcoR1. Now there are two more sites that is written as AMP, that is ampicillin, and TET, that is tetracycline. So ampicillin and tetracycline, these are nothing but the antibiotic resistance sites. So if you are targeting any one of the antibiotic resistance site, and say for example you have chosen the EcoR1 plate, EcoR1 ko you have removed and you have added your own particular gene. Once you are adding your own particular gene, it will destruct the ampicillin site. And ampicillin site, once it is destructed, what will happen? If you grow it on ampicillin, it will not grow. It will not grow. Why it won't grow? Because ampicillin resistance has been removed. Ampicillin resistance is removed and antibiotic will not allow it to grow. Whereas on tetracycline, what will happen? There will be growth because the resistance is still valid and it can still grow onto that antibiotic. So even this is being used as a plasmid, but it has a disadvantage, that is it will have two sets of resistance, and because of which, that is one that is ampicillin, other that is tetracycline. Because of the two things, we need to have two-step procedure, यानी एक साथ simultaneously two plates needs to be produced, that is one for ampicillin, one for tetracycline, and one for the model which you will be using for comparison. Okay, because you need to compare that this particular colony is not growing on that particular antibiotic site, so that means that has been evolved. So that kind of thing, that is a very long taking procedure. So majorly it's not advisable to use PBR322. Instead of that, a major change has been evolved with using of PUC8, that is having advantage कि उसका higher copy number है and it can be easily identified with a single step procedure. Why? Because it is going to have the LAC-Z gene. This LAC-Z gene is the chromogenic gene. That means it is going to produce color. So blue color is being given to the colonies which will have this gene. So blue colored colonies means these are the wild type of colonies. These are the non-recombinant colonies and these are not desirable. If they are colorless, then colorless are going to be the ones that are wanted, or these are the ones that you desire. So colorless wale ko you can directly choose, and the blue colored colonies that you would see onto a plate, that you won't choose. So based on that, you can easily classify the recombinant and the combinant ones. So वो एक ही step में procedure will get completed, and that would help you to save much more time. So you don't need to sit and carry 4 plates around you. Just in one plate, you would complete the correction about what is combinant, what is not combinant. So after we have seen the different kinds of tools about like कि cut करना है, paste करना है, multiply करना है, and even to transport it to the host cell. Now once we have all the ingredients together, now we can see the actual procedure. जैसे सब इकट्ठा कर लिया, all the tools are ready with us. Now let us go and create the recombinant DNA. So for the recombinant DNA procedures, first of all, you need to isolate the DNA. Isolate DNA यानी plasmid जो भी है, that plasmid में से पूरा का पूरा DNA बाहर आना चाहिए. Whatever the plasmid you are choosing, for example, सबसे पहले जो बनाया गया था, it was coming from Salmonella. So the plasmid was used that was from Salmonella. So Salmonella का plasmid isolate करते आना चाहिए. Then you can find out what is desired by you. So once the desired site has been chosen, the desired site has been got. अब ये desired site को cut करना है. So that will be the fragmentation of DNA by the restriction endonucleases, the second step. Once it has been fragmented, it has been cut, you need to separate it क्योंकि you just need to produce that particular desired DNA fragment. So उसको separate करते आना चाहिए from the one that you don't need. The black one is the part that you don't need, so remove it off. And the red colored part is the one that you need. So that is the part you will carry forward to the fourth step, that is amplification. अब ये एक ही जीन से तो कुछ नहीं होगा because you need much quantities, much more trials. So you need to amplify that. So for the amplification, what you are going to use? You are going to use the technique that is called as PCR. So once the PCR say amplification has been carried out, after amplification, you need to paste that particular thing into a new vector. So अब ये vector आ गया हमारा vehicle. On this vector, you need to paste your amplified DNA segment. Once you have pasted the amplified DNA segment, you need to transfer it to the host cell. होस्ट सेल के अंदर आपने इसको ट्रांसफर कर दिया. So once you have transferred it to the host cell, now it needs to culture. अब ये host cell को क्या करोगे? उसको खाओ, पिलाओ, सारे nutrients खिलाओ, and now it will start to divide. And once the host cell starts to divide, what it will have? It is going to have the DNA segment that you wanted, the DNA segment that you have cultured, the red color part that you need. So अभी एक से host cell के अंदर how many you have 3 of. So अब ये 3 से dot 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 में कितने बन जाएंगे? लाखों करोड़ों number of genes will be obtained. Now once these cells have been obtained, अब ये cells कैसे होंगे? These cells will occur as a pellet. ये किस form में होंगे? This will be in the pellet form. Now from the pellet form, you need to separate it, you need to extract it. You need to extract the product. You don't need the entire cell culture, you just need the desired product. For example, the gene that you were using, okay, the gene that you were using, that was for the antibiotic. So आप क्या कर रहे थे? You were producing the antibiotic. So gene antibiotic production के लिए था. आपने gene को क्यों modify किया? Because you were interested in the product. आपको product चाहिए. That product is nothing but your antibiotic. Antibiotic को आपको क्या करना पड़ेगा? You need to extract it. So this extraction will be the last procedure, that is your desired product. यानी आपने ये particular host cell से वो antibiotic का production करवाया. That was the production unit, and extraction will be done, and then will be the packaging stuff. So antibiotic will be prepared not by the human or not by combining each and every bacteria को change करके. Instead of that, एक में changes करा, और जैसे उनकी colonies बढ़ेंगे, your product will also multiply along with it. So that is nothing but the beauty about the recombinant DNA technology. The next is to understand each and every step in detail. अभी तक जो बताया, that was just in brief. अब ये एग्जैक्टली कैसे सब होगा दैट वी नीड टू अंडरस्टैंड. सो आइसोलेट द जेनेटिक मटेरियल. हमने जो सबसे पहले एक एग्जांपल देखा था दैट वाज फ्रॉम द साल्मोनेला. सो साल्मोनेला में से हमने जो प्लाज्मिड को निकाला है, हाउ आर वी गोइंग टू रिमूव इट? Not necessary that we are always using the salmonella, that is a bacteria. It might happen that we are using a fungus cell, a plant cell, an animal cell. You don't know, anywhere you can find the desired gene. So for that, अब साल्मोनेला में से निकालना है. सो साल्मोनेला की ज़रूरत है. सो फर्स्ट यू नीड टू कल्चर द बैक्टीरिया दैट यू वांट. Okay, so not necessary always you need the bacteria. It might be any other cell that you wish, but you need to culture it, you need to harvest it. Once it has been observed in a good amount of number, you need to break open the cells. अब cells को break करना है because bacterial cells will have their covering. So those coverings need to be broken down. So bacterial cell को आप किससे break करोगे? Lysosome से. Fungal cells will have their fungal wall that will be broken by chitinase, and plant cell wall having cellulose in its cell wall will be broken down by the cellulase. Once it has been opened to the protoplasm, now you have to extract the DNA. अब extraction of DNA के लिए आप क्या कर सकते हो? एक तो सिर्फ DNA को निकालो, या बाकी सबको खत्म करो तो सिर्फ DNA बच जाएगा. So the second option is much more feasible in the practical state. So instead of just removing the DNA, just remove everything else. Whatever is left is nothing but the DNA. So once it has been completed, the purification of the DNA should occur, and that purification will be done by addition of chilled ethanol. Now this is a very important part, how you are going to extract the DNA. So DNA extraction will be done by chilled ethanol, and you are going to remove that from the suspension with the help of a spooler. So after adding the chilled ethanol, यहाँ पे एक pellet form जैसे होगा, and this will be nothing but called as spooling. With the help of spooling, you are going to remove that DNA, right? So spooling will be done by a spooler. This is kind of a metal ring. With the metal ring, you are going to remove that DNA, the purified one. After the isolation of the DNA from the bacteria or any cell that you are wishing, second step will be the fragmentation of it. Only the desired part that you want. So restriction enzymes को आपने ये DNA के साथ छोड़ दिया at particular temperature, that is 37 degrees Celsius. You also have to maintain कि जो cell है वो cell जैसे ही same conditions maintain हो in the artificial world as well. So pH and water content should be taken care of, and all this you also need to provide the nutrition for it. So how can you check if exactly the fragmentation of DNA is occurring or not? So you can use the agarose gel electrophoresis method. So using this electrophoresis, you can exactly see कि कितना fragmentation हुआ है, and based on that, if you want to stop the process or you have to wait for it, that you can decide. So very important part to remember about the fragmentation is कि आपने जो भी fragmentation का endonuclease choose किया है, the same scissors, that is same molecular scissors, needs to be used to cut the vector as well. So अगर आपने like using salmonella, if you are using salmonella, so salmonella का DNA को cut करने के लिए if you have chosen EcoR1, तो EcoR1 should only be used to cut the vector, that is from the E. coli. So E. coli और salmonella दोनों को आप किससे कट करोगे? EcoR1 से. Same restriction enzyme need to be used. The third step is separation. आपने जो कट किया है आपको उसको separate करके उतना ही part आपके pocket में रखना है. So separation will be done by the electrophoresis method. So as you know that DNA are going to be the negatively charged. So जो अगर आपने उसको current pass करवाया है with the positive and the negative terminals, so what will happen? The negative will be attracted to the positive charged particles. So the negatively charged DNA segments will try to move from which position to which position. It will start moving towards the positive electrode, that is anode. So it will move from cathode to anode. Cathode is the negative position. Negative position say anode, the positive electrode way the DNA are going to move. So these fragments, how are they going to move? They will be moving according to their size. So smaller the fragment, farther it will move. जितना छोटा है उतना दूर जाएगा. So here in the GIF also you can understand that if it is a large sample, वो कम distance travel करेगा. But if it is a small sample, very small quantity of fragments are there, then it will travel more closer, more farther away from the negative terminal. So based on the, this all things that you have done to the separation, now you are not going to see that with your naked eye. DNA segments बहुत छोटे होंगे and you cannot see them that easily. So to visualize them, you need to stain them. So staining किससे करोगे? Staining करोगे ethidium bromide से. Once you are using ethidium bromide, under the UV radiations, you can visualize them as bright orange bands. Bright orange bands, here you have been provided a photograph that can show you how the bright orange bands are seen. The bright orange bands के side में you are going to place a scale, और ये scale पे there will be markings based on the base pairs. Say for example, this particular area is 300 kilo base pairs. This area is going to have some other base pair scaling. So based on what kind of a gene that you want, you are going to select the base pair scaling, and that particular gene is going to be isolated. So isolation ke liye what you will do, you are going to cut that particular section from the agarose gel, and this extraction process is nothing but called as illusion. So illusion का definition पूछा जा सकता है. It is separation of the DNA from cutting it with the agarose gel and extracting the gel piece. That is nothing but illusion. So after this, once you have separated and isolated, the fourth step is going to be the amplification. So amplification of gene that you have already separated and you have also isolated it. Now isolated gene needs to be multiplied into many times. So the PCR is going to be used. So PCR में 3 steps होंगे which are temperature dependent. So you need to take care कि जो भी आप steps use कर रहे हो, that temperature is maintained, otherwise it is not going to give you the result that you want. So first step is denaturation. Denaturation is making the bonds broken. The bonds break करने हैं, that means you need to heat that particular solution at 94 degrees Celsius. Once you have heated it, there will be bonds that are broken. After the bonds are broken, you have to suddenly cool the mixture at about 50 to 60 degrees Celsius, and from that the primers will bind. That is the process of annealing. And after the primers have been bound, primers क्या करेंगे? They would be the markers which will help the next procedure to be, it will guide the DNA polymerase to proceed making multiple copies. So primer extension or polymerization will be the next step of the amplification, that is at 72 degrees Celsius. And after the DNA polymerase, again it is an enzyme जो एक template से multiple बनाएगा copies. So that will be done in the amplification procedure using PCR. So how can you make 1 billion copies of the desired gene that you want? So 1 billion copies produce करने के लिए how many PCR cycles do you need? So that can be calculated based on the formula, that is 2 raised to n, wherein n is the number of cycles that wherein you are repeating the PCR. So 1 billion copies seems that is a huge amount of number of copies, but for 1 billion copies you just need how many cycles? 30 cycles. In 30 cycles of PCR, you are going to obtain 1 billion copies. So that is the beauty about the PCR कि एक strand से 1 billion तक पहुंचने के लिए you just need 30 PCR cycles. The next we need to see where you can use this PCR. अपने PCR का principle पढ़ लिया, you have understood how does it work, but now the question is where does it work and how can you use it? So you can use it in the diagnosis of pathogens. Just for example, in COVID days, you needed to understand if the patient was positive or negative. So you had done the test that was called as RT-PCR. So that is a kind of a PCR wherein you are seeing that you are diagnosing whether the patient is positive for the pathogens or not. Sometimes it can also be used for specific mutations. For example, कोई अगर genetic disorder है, for the genetic disorders you can have the PCR done to understand, क्योंकि अगर multiple copies वो particular gene का बनेगा, then that particular will have multiple copies, और जब आप उसको identify करने जाओगे then you can know कि because of the multiple copies वहाँ पे उसका density बढ़ जाएगा. So density बढ़ जाएगा, that means that particular thing is positive for the one which you have carried out the PCR. So that is the application of this PCR technique. The next is ligation. Once you have amplified 1 billion copies बना लिया, वो बने हुए copies का क्या करोगे? You need to paste it to the vector. वेक्टर को पेस्ट करने के लिए you are going to use the DNA ligase, and once it is ligated with the vector, now you would go for the further cloning. So the sixth step will be insertion of the recombinant DNA. Where you need to insert it? You need to insert it into the host cell. Once your vehicle is ready with the passenger, it needs to reach to the destination, that is the host cell. So now it will reach via different ways. So there can be 5 ways by which you can take the transporting vehicle into the host cell. So the first method is transformation, wherein you are going to use the heat shock method. यानी आप उसको एक गरम करोगे और सडनली आप उसको ठंडा करोगे. So 42 डिग्री सेल्सियस पे जब सेल्स को आप गर्म करोगे और सडनली once you place it on ice, that will be a sudden temperature shock, जिसकी वजह से the plasma membrane will increase its permeability, और permeability increase होने की वजह से your vector will get entry into the host cell. That is the first technique. Second you can use is micro injection. Micro injection यानी छोटी सी needle है, वो needle के अंदर you will place your vector that is having your recombinant DNA fragment, and you will directly inject it into the nucleus. So here you just need to locate the nucleus and just inject it into that nucleus. So it seems very easy, but you consider the size of the nucleus, consider the size of the vector. All this thing needs to be done under the microscope with large magnification so that you can see whether your needle is injecting into the nucleus only or not. The third method can be the gene gun. So very important part to remember, the difference between micro injection and the gene gun is micro injection जो होता है, it is used for the animal cell. Animal cells में you will use micro injections, whereas the gene gun or the biolistics will be used for the plant cell. Biolistic या gene gun क्या होता है? This is the DNA fragment. अब ये DNA fragment को you will coat it with either gold or you will coat it with tungsten. Once you are coating it, these will be bombarded with a high velocity. जब ये high velocity से जाएंगे, it will be getting entry into the cell because वे इतने fast जाएंगे कि the cell membrane won't understand what happened. So that is a technique for the plant cells. The next one you can use is disarmed pathogens. यानी pathogens का जो pathogenic या virulent factor है, virulent factor will be targeted, और virulent factor की जगह पे you will insert your desired gene. Virulent factor के जगह पे you will insert your desired gene, and once it will enter into the cell, then it will transport the desired gene instead of causing infections. So such kind of pathogens, for example, the agrobacterium or retroviruses can be used for the disarmed pathogens. And the last technique is going to be electroperforation, wherein instead of heat you would be giving short high field strengths, यानी आप electricity use करोगे, short impulses will be provided, because of which the permeability will increase. So these are the 5 ways by which the host cell can give more permission for the entry of the recombinant DNA. Now after the entry of the recombinant DNA, अब host cell को आपको culture करना है. अब जो host cell आपने choose किया है, for example the bacteria that you have chosen, अब bacteria के अंदर जो आपको चाहिए था, whatever you needed, वो जो plasmid था, the circular one, अब वो plasmid के अंदर already your desired gene is located. Now this desired gene needs to be in multiple copies of the bacteria. हर bacteria के अंदर you need that gene. So for that you need to culture the bacteria so that bacteria has the desirable conditions to multiply itself. So nutrient medium will be provided, और ये आपको कुछ ऐसे you don't have to make it for 1 liter 2 liters, you need to make it at a very huge quantities. So huge quantities में बनाने के लिए you would be using the machine that is called as fermenter, or also called as bioreactors. In the fermenters or the bioreactors you have to carry out the process of culturing of host cells. तो ये कुछ 1 किलो 2 किलो नहीं होगा, that would be in hundreds of kgs of the host cell. And for that reason, once you are carrying out this procedure, you need to take care कि वहाँ पे aseptic conditions हो. That means there shouldn't be any other kind of bacteria or any other infective material into that host cell culture. Second, very important, supply of oxygen. Every living cell needs oxygen. So oxygen provide करना ज़रूरी है, because you need to have to give every optimum condition. The best of the best should be provided so that you can have many more divisions of the host cell. So oxygen provide करेंगे with small bubbles. Why not large bubbles? Large bubbles होंगे तो हो सकता है that the cell might get disrupt when the bubble pops out. So small bubbles में you would release oxygen, and for that a sparger is used. Now what is a sparger? Sparger is nothing but a long tube, and the long tube will have small pores, and from the small pores there will be the exit of the oxygen, so that won't give you large bubbles but small bubbles of the oxygen. That is a sparger. The third important thing that you need to continuously stir the mixture. Stirring is very important because the host cell should not go and stick onto the bottom. क्योंकि अगर bottom में stick हो जाएंगे, there is a possibility that the lowermost cell, because tankers, these are very big vessels. Okay, अगर जो नीचे के host cells है, हो सकता है they won't get oxygen because they are sitting very lower at their age, sparger ऊपर है. So for that what you will use a stirrer, so that continuously the cells are in motion, and because of the motion what will happen, जब आप नींबू पानी बनाते हो, जूस बनाते हो, once you start stirring, what will happen, there will be foam creation, या बुडबुडे बनना शुरू करेगा. So there might be some agents, that is anti-foaming agents, needs to be used, so that once you are adding air or once you are stirring the mixture, it shouldn't create foam, because you don't need the foam, so anti-foaming agents needs to be added into it. Fourth step is you need to have proper heat circulation. So जब आप stirring कर रहे हो या once you are adding oxygen, the heat will be released. जैसे आपको sweating होता है, perspiration होता है to release the heat, same way there might be some technique जहाँ से ये cells जब grow कर रहे हैं, their energy is going to produce heat. Now this dissipated heat by your culture needs to be thrown out. नहीं तो क्या होगा, if the heat is not thrown out, if heat is there only, it will increase the temperature, and temperature should not increase. So to keep the temperature below, there needs to be what? There needs to be continuous circulation of cold water. So circulation of cold water will provide the heat dissipation, and that will be allowing the microbes to grow in the proper temperature. Finally, सबको खाना ज़रूरी है, so that nutrients needs to be provided. pH needs to be taken care of. Oxygen you are already providing. So with these 5 requirements in the fermenter, you can have the culturing of the host cells. Now just we have understood about what needs to be provided, but what is this vessel that we have seen, that is the fermenter. So this fermenter, let us see it in detail. So fermenter जो होता है, the fermenter is going to have a stirrer. Stirrer है यानी ये kind of a spoon है, a big spoon which is going to be mixing the oxygen and the culture. So the nutrients, the oxygen supply is all evened out during the culture procedure. So in this, the bioreactor system is also installed in some kind of bioreactors. यहाँ पे you can see the amount of oxygen delivered, the pH, the temperature, the foam can be assessed. Everything can be assessed externally without opening the fermenter, and that kind of machines are available in two kinds, that is simple stirrer tank bioreactor or sparged stirrer tank reactor. So you can have the spargers creating small bubbles, so increased amount of oxygen can be used, या basic वाला भी आप use कर सकते हो for small scale bioreactors. So there are different kinds of reactors, the fermenters that can be used. Now there are two kinds of fermentation procedures that is occurring into this. So first one you can use, that is the closed system or batch fermentation. Batch fermentation यानी आपने एक कल्चर को अंदर डाला, you have added one culture, and one culture का जब भी product form हो जाएगा, after the product is formed you will remove it, and next it will be clean and then it would be used again. So that is one time use. So it is a closed system. एक बार अंदर डाल दिया, you are not going to open it, and you will keep it as it is until the product has been formed. So for example, you can correlate it, this batch fermentation, in the form of say wine production. Wine production में barrels होते हैं. Barrels will be kept for say 5 years, 10 years. Will not touch it. We'll keep it as it is in a closed condition. एक बार आपने grape juice डाल दिया है, fermentation will start. You will not add anything inside it. Okay, new nutrients will be added. Once it is added, everything, it is closed, it will be kept as it is. Even the same thing can be used for cheese as well. The aging of the cheese, that is also kind of a batch fermentation, a closed system. Whereas the opposite of it, wherein you will be adding, you will be removing the product. You will add it, remove it. That will be the simultaneous procedure. Add and remove will be done at the same time. Okay, so this kind of procedure is called as a continuous culture, or it is the open system. यानी एक साइड से आप fresh medium add करोगे, और second side से you would be removing or you would be draining the desired quantities. So such kind of continuous culture is also used in curd making. Commercial curd making जो होता है, you would be adding milk from above and नीचे से curd will be extracted. So that is the continuous culture. So these are the two different kinds of fermenters which are used, the batch fermenters or the continuous fermenters. The last step, once you have obtained your culture colonies, you have obtained the fermented process. Last is extraction. जो आपने जीन से बनवाया है, जो भी जीन था, say for example antibiotic production gene you have inserted. Okay, antibiotic gene has been inserted. You need, what do you desire from it? The process is nothing but the antibiotic. So this antibiotic needs to be extracted. Now this extraction can be done by a process that is called as downstream processing. Downstream processing, wherein you will be recovering and purifying the product that you need. So once the content of the fermenter has been collected, जो भी cells थे सबको collect कर लिया, you have done the filtration, the centrifugation, purification is being done कि प्योरेस्ट ऑफ द प्योर एंटीबायोटिक आपको मिले. हो सकता है एंटीबायोटिक्स नहीं यूज कर रहे हो, यू आर यूजिंग इट से फॉर हॉर्मोन्स, you are using it for enzymes, or you might be using it for some other important things that you want to make. या हो सकता है वैक्सीन प्रोडक्शन हो, you can use it for n number of quantities of times. So these all things need to be purified so that the best is available. So purification hoga. After the purification, you need to formulate with some suitable preservatives, because these are not going to be used. You need to market them. You need to sell them. So yes, selling ke liye what you will do, you are going to add preservatives, and then after adding preservatives, you will get, say for example, one bottle of vaccine. अब ये एक bottle of vaccine को you need to undergo the clinical trials. So clinical trials you will make it available on the lab animals. Lab animals पे आप टेस्ट करोगे कि जो भी प्रोडक्ट आपने बनाया है whether it is suitable for the humans or not. Once it check pass the lab animals, then you can say the human consumption is okay, then it will be sold out in the market for the human use. So with this we can understand the entire process कि आपने जीन को कैसे मॉडिफाई किया, आपने उसको कैसे बनाया, what were the tools that you used to form this particular desired product using the bacteria, why you aren't using the conventional methods. This all things we will understand in the next chapter about the application of it. This was just to introduce how the procedure is followed out. अब ये procedures कहाँ पे use होंगे in real life, that we'll be seeing in the next chapter. So for now, we are covering up the procedure of this recombinant DNA formation. So with this, we will come back soon with the next chapter wherein you will understand that where and how in real life, in real world, this recombinant DNA technology was used. So it might be used in agriculture, it might be used in medicine. बहुत सारी fields है जहाँ पे इसको use किया जा सकता है. And to know more about how it was used and what are the products that are actually made from this that you are using right now as well. So for that, stay connected to Biophilia. Don't forget to like, share and subscribe. Until next time we meet again. Bye bye everyone. Thank you.